It's hard to believe, but there are only 3 talks left. The first one will be delivered by a young scientist from VIB in Belgium. Again, let's first see the video of the institute.
At VIB, we're shaping the future of life sciences. Our scientists push the boundaries of knowledge to combat human disease. We apply biotechnology to strengthen global food security. From fighting antibiotic resistance to producing next-gen medicines, microbes are part of the solution. With world-class technology and data-driven approaches, our teams deliver results that matter. At VIB, science is more than research. It's commitment to improving lives and building a better future. Driven by curiosity, powered by collaboration, focused on impact. That's VIB.
Seeing clearly can save lives. Pauline Bardet will present separate-seq and a new way to distinguish tumor tissue from healthy lung, and thus bring clarity to cancer research. Please welcome Pauline. Let's try to connect with your fun base at home in Belgium. Hello guys, very nice transparency. Thank you very much for following and for your support for Pauline. Great, I think everything is ready. Take your time, 4 minutes.
Great. Let's start with a movie. Sorry, this was not planned. Maybe it was. Anyways, if you look on the screen now, I guess if you squint your eyes, it looks like a lung, right? A lung with cancer. Now, unfortunately, the image is very blurry, and it's going to stay that way. Why? Because as cancer researchers or cancer immunologists, this is the image we're looking at in a lab. We're using what we call mouse models to mimic human cancer, but in mice. Unfortunately, there's a lot of issues with these models. In the case of lung cancer, a lot of these models actually develop tumors all over the lung, with boundaries that are very unclear, which makes that when I am in the lab, I have to analyze the whole lung instead of just analyzing a tumor, because I cannot separate it out, which makes a very blurry and messed up sample. Now, in the clinic, doctors are actually able to very precisely excise tumors from patients' lungs, from the rest of the healthy tissue. And I was wondering if I could do the same in a mouse model. And after a whole bunch of different optimizations, this took about half of my PhD. Yes, you guys all know. I finally got to that model, a model where the tumors grow in very clear, precise locations, so that I can actually, just like with patient biopsies, cut the tumor out and isolate healthy and tumoral lung separately. So what I can now do is zoom in onto the tumor and get a clearer picture of what is happening in the tumor, how the immune cells in the tumor are different from those in the healthy tissue. Unfortunately, you can see that it's clearer, but there's still a bit of static remaining on my TV. And that is because some of the immune cells that we're actually identifying are present in the blood vessels or in the vasculature of our tumors, and they're causing some interference on the signal. And that's where separate-seq comes in. It's a technique I developed during my PhD, which allows you to tag the immune cells present in the blood vessels alone and identify them further with single-cell RNA sequencing. This way, you can distinguish the signal from those in the blood vessels to those actually present in the tissue. Now, the image is suddenly a lot clearer, and we can actually start separating our signal very clearly. Firstly, we have the noise from the adjacent and the tumor tissue, which has now been split by using the model. Secondly, by really identifying the cells that are present specifically within the tumor tissue. Now, let's take, for example, neutrophils, one of my favorite cell types. Now, I identified a whole bunch of them in our model, as you can see here. I'm just going to call them by their colors to make it a bit simpler. But if we step back and we zoom out and we start actually separating them, we can see that those in the adjacent or healthy tissue are mostly of the yellow type. They all look very similar, whether they're in the tissue or in the vasculature of that healthy tissue. If we go into the vasculature of the tumor, they're still all mostly yellow. But then the picture shifts completely when we enter the tissue of the tumor. You can see that the neutrophils have completely adapted. They're not all yellow anymore. They took on a lot of different colors and flavors. And this we wouldn't have known, we wouldn't have appreciated if we wouldn't have made those distinctions. Now we know that we have to look into those specifically present inside of the tumor tissue. And this way, we're starting to tune in to a clearer picture of lung cancer. And by doing so, I hope that we can actually start to change the story. Thank you.
Thank you very much, Pauline, for your excellent presentation. Thanks a lot.